Today we're talking about Chapter 8, Section 7, and this is the topic of mutations. Mutations are any type of change in the genetic material of a cell, and there's really two categories that we're concerned with. The first one is called gene mutations because that involves an actual change to the nucleotide sequence. This could be a change to the DNA in a cell, or it could be an error that happens during transcription where the incorrect mRNA is made from a piece of DNA. The other type of mutation is called a chromosomal mutation, and that's going to involve a much larger scale mistake in the actual structure of the chromosome itself, where a piece of it may break off, or maybe during the process of cell division, you have the wrong chromosome number end up in a daughter cell. Those are normally very serious mutations, and those are not really the ones that we're going to focus on. Most of what we're focusing on here would be those point mutations or single changes in a nucleotide of a DNA molecule, like what you see pictured here on the left. So let's talk about those types of genetic mutations in a little more detail. The first one is called a point mutation. A point mutation, like the name suggests, is a mistake at one particular point in a strand of DNA. And so what happens is if DNA is copying itself and there is a substitution or putting in a base that's incorrect in place of one that should have been there, that is the most common way that you're going to get a point mutation in your DNA. Normally, this is going to result in a protein that has an incorrect amino acid added to it, because if you think about what the purpose of DNA is, it's to have the correct sequence. So when you make mRNA from it, the mRNA contains the code for amino acids. So if you have a mistake in one of your bases, you're going to make the wrong mRNA, and then often that will result in the incorrect amino acid of your protein. Sometimes, because there are multiple codons that would code for the same amino acid, you could have no change at all in your protein. And if this is the case, we say that it's a silent mutation. Yes, it was a mistake, so it is still a mutation, but the overall protein that's produced would be the same. So a person who had a mutation like this would never even know that they had it. So here is a comparison between a normal strand of DNA on the left and the strand of mRNA that it would produce through transcription. And then you can see that mRNA, which is split into codons, is translated into those 4 amino acids you see at the bottom. Now, over on the right-hand side, in this DNA strand, we have a mutation. There is a mutated base right after the T. In our mutated strand, it is an A. Over here, in our first strand, it was supposed to be a C. But now that we have that mutation and have placed an A in the DNA strand instead, when it undergoes transcription, now this in the mRNA will become a U. So GAU is the code for the amino acid ASP. We originally had the codon GAG, and that was the code for the amino acid GLU. So only one change happened. We had a single point mutation for one particular nucleotide base, and yet that resulted in a different amino acid being added to the resulting protein. Now, the other type of genetic mutation you could have is a little bit more serious. We call this a frame shift mutation because it's disrupting the reading frame. And what we mean by reading frame is just the group of 3 bases, how a ribosome only reads 3 bases at a time. That is the reading frame of the ribosome. And this is caused anytime you change the number of nucleotides. You might insert an extra base, or maybe you delete a base, so a base would be lost from the overall sequence. Changing the number like that is going to change the way that the groups of 3 are read by the ribosome. So let's look at a picture of that. Again, we have our normal DNA sequence on the left and our frame shift mutation on the right. Now we have a deleted base. Right here, after the A, it was originally AGG, but now you can see that first G has been deleted, and now it says AGC, and then the rest of the sequence. So the codon was supposed to be UCC, and we still have this first U, but then we don't have that second C because that G was deleted in the DNA. So everything is going to shift over. What used to be our third base in the codon is now the second, and then what used to be the first base in the next codon is now our third base here. So the codon UCC is now going to be read as UCG, and this is the code for a different amino acid. Now, because this causes the shifting of the entire reading frame, that means every single codon from this point on is going to be misread by the ribosome, and this is almost always going to make a non-functional protein. So let's take a look at that in a little bit more detail. We have our same sequence here. This is an mRNA sequence, and this picture is highlighting what would happen if you had two different reading frames. The original reading frame number 1, is shown at the top. Now let's say we have a deletion. Let's say we delete that first base. Well, now that is going to shift our reading frame. So go ahead and pause the video here. See if you can determine, based on your genetic code chart, what the new sequence of 3 amino acids would be that's based on this reading frame number 2, and then you can play the video once you have your answer and check your answer. All right, hopefully you've had enough time to figure that out. Our reading frame 2 is going to produce this sequence of amino acids. First is going to be ASP. The second one is THR. And the third amino acid will be VAL. Now there are going to be more amino acids because this is just a segment of a much longer strand of nucleotides, but obviously we're not given the third nucleotide here in the codon, so we are unable to continue any farther. But you can definitely see, even just from these first few amino acids, that our original reading frame gives us a very different sequence compared to reading frame number 2 when we had a deletion. So this is normally going to be a very serious mutation. Again, it's probably producing a protein that is completely non-functional, and this would be the reason why we have a lot of the genetic diseases that exist today. Now, we normally think of mutations as being bad, and most of the time they are, but they can actually be beneficial as well. So when a mutation happens and what the result of that mutation is really going to determine if it continues in a population or not. So first of all, this probably is very intuitive. Think about a mutation in a body cell, like your skin cell. That's not really a mutation that you are going to pass down to your offspring. It might affect you as an individual. For example, if there's a mutation in your skin cells that cause them to divide uncontrollably, that would be skin cancer. And you as an individual would definitely have to deal with the effect of that mutation, but you're not going to pass that down to your offspring. Mutations in sex cells, however, we call these the gametes, the egg cells or the sperm cells. They can affect your offspring. So if there is an error in copying the DNA before the process of producing the egg and the sperm cells, then those cells could contain mutations that would be inherited by the offspring. So if we do have mutations that can be inherited by offspring, those mutations that are bad are probably not going to allow the offspring to live its best life. They're probably not going to be passed down as often as beneficial mutations. If there is a beneficial mutation and the offspring can inherit it, the offspring would then have some sort of benefit, some kind of adaptation that helps them survive, and they're likely to pass that down to their offspring as well. This is really the basis of natural selection, and we'll talk more about that in Unit 9, which is evolution. Now, mutations can be caused by several different factors, so let's talk about what those factors actually are. The most common way that DNA can become mutated is during the process of replication. And that makes sense if you think about what DNA replication involves. It involves a very long strand of DNA, lots of different nucleotide bases, and it happens very quickly. So statistically speaking, it's likely that when a replication or when an error occurs, it will occur during the process of replication. And this can be just random mutations. It can happen at any point along a DNA strand. There's nothing that really causes it. It's just random chance when DNA copies itself. There are, however, some things that can induce mutations or make it more common for a mutation to happen. We call those factors mutagens. A mutagen is anything that increases the chance of that mutation occurring. The most common mutagen that you probably are aware of already is radiation. Any type of high energy radiation like UV rays from the sun or X-rays. Those are very damaging to the DNA in the cells. They can cause mutations, and that's why it's important to do things like wear sunscreen. Or if you have to get an X-ray, you wear a lead shield that protects parts of your body that aren't being X-rayed, because those X-rays can definitely cause mutations in your DNA. The other type of mutagen would be some sort of a chemical mutagen, and that might be things like mustard gas or sodium azide, which used to be used in car airbags. It's no longer used in airbags. They have modern replacements for those. But there are some pretty nasty chemicals out there. We normally try to avoid them. But if they have any type of interaction with your DNA and can damage DNA, then they would be classified as a mutagen. And a lot of times those mutations lead to cancer. So if you've ever heard the term carcinogen, normally that's a type of mutagen that leads to the specific mutation that would give a person cancer. So that's going to be it for our discussion about mutations today. Hopefully you found this information to be helpful. And if you have any questions, please make sure that you ask them in class.